Welcome to the TOEFL listening section. In this part, you will hear one or more conversations in an academic or campus context, such as a discussion between a student and a professor or between two students. You may take notes while listening. Listen to part of a conversation. You will hear it only once, so listen carefully. After the conversation, you will answer questions about what you heard. Some questions will ask you about main ideas and details. Others will ask about implied meaning, speaker attitude, or how ideas relate to one another. Use both what is stated and what is implied to answer.
Good afternoon, Dr. Carter. Thanks for meeting with me. I wanted to discuss my project on microplastic health impacts and get your advice on study design.
Hello, Lena. Of course. Microplastic health impacts is a timely toxicology topic. Tell me the focus you have in mind.
I want to look at biological effects of plastic particle ingestion, especially how particles affect the gut and whether they translocate to other organs.
That is central. Ingestion is a major exposure route for humans. We know microplastics have been detected in human stool and even in placenta samples in recent studies, so addressing gut effects and translocation is important.
Right. I read that ingestion occurs through seafood, bottled water, and even dust on produce. But I am unsure which endpoints to measure in vitro and in vivo.
You should include endpoints that reflect both physical and chemical toxicity. For physical effects, think about epithelial barrier integrity, tight junction proteins, and particle-driven inflammation. For chemical effects, consider leached additives and sorbed pollutants like phthalates or PCBs that can desorb from particle surfaces.
So measuring tight junction proteins and cytokine expression makes sense. What about oxidative stress and genotoxicity?
Yes. Reactive oxygen species generation, DNA strand breaks, and markers of genotoxicity are relevant. Several studies using intestinal cell lines showed increased ROSIS and altered expression of antioxidant enzymes after exposure to polystyrene nanoparticles.
I also want to understand translocation. How likely are microplastics to cross the gut barrier and reach organs like the liver?
Translocation depends on size, surface chemistry, and the presence of a protein corona. Nanoplastics under a few hundred nanometers are more likely to cross enterocytes or be taken up by M cells in Peyer patches. Experimental mouse models have shown polystyrene particles reaching the liver and inducing inflammatory responses.
You mentioned the protein corona. Can you explain its role in uptake and toxicity?
When particles enter biological fluids, they acquire a layer of proteins and biomolecules. That corona changes the effective surface properties and can increase cellular uptake or alter immune recognition. Weathered environmental particles may have different coronas which change interactions with gut cells.
That helps. For in vivo work, should I use pristine laboratory polystyrene beads or environmentally aged particles?
You should include both. Pristine particles are useful for mechanistic clarity, but aged particles with sorbed contaminants and biofilms reflect real-world exposures. Comparing them will clarify how environmental conditioning modifies toxicity.
A big challenge I see is dose selection. Many studies use very high doses. How do we choose realistic exposure levels?
We need to anchor doses to measured human exposure estimates. Use concentrations from bottled water and seafood studies as a starting point. Then include a higher dose to explore dose response, while noting that many laboratory doses exceed environmental levels.
That makes sense. Do you think chronic low-dose exposures are more relevant than acute high doses?
Chronic low-dose exposures are likely more representative of human experience. They may reveal subtle effects on the gut microbiome, low-grade inflammation, and endocrine disruption from additive leaching that acute studies miss.
You mentioned the microbiome. How do microplastics alter microbial communities?
Particles can act as substrates for biofilm formation and selectively enrich bacteria. In animal studies, microplastic exposure altered diversity and metabolic output of gut microbiota, which can influence immune function and metabolic endpoints.
Are there specific human health outcomes linked to these changes yet?
Direct causal links in humans are not established. We have mechanistic animal and cellular evidence suggesting pathways to inflammation, metabolic dysregulation, and endocrine disruption. But epidemiological data are limited and confounded by many factors.
What about vulnerable populations? Should I consider pregnant animals or infants in the study?
Yes. Pregnant animals are important because some studies indicate particles can reach the placenta and potentially the fetus. Infants are vulnerable due to higher intake per body weight, and feeding bottle microplastic studies, including developmental windows, will strengthen relevance.
For analytical methods, I was thinking of using fluorescence-labeled particles for tracing. Any caveats?
Fluorescent labeling is useful but can mislead if dye leaches. Validate with particle-specific techniques like Raman spectroscopy or pyrolysis GCMS to confirm plastic identity. Also characterize size distribution, surface charge, and any sorbed chemicals.
I will include those characterizations. For endpoints, I will measure ROS cytokines, tight junction proteins, microbiome composition, and check liver tissue for particles. Does that sound comprehensive?
That is a solid plan. Ensure you include appropriate controls and consider time points that capture acute uptake and longer-term accumulation. Also plan for statistical power given variability in biological endpoints.
One last question. How do we communicate uncertainty to non-specialist audiences when discussing health impacts?
Be transparent. Emphasize mechanistic findings and the difference between experimental doses and real-world exposure. Highlight known pathways, such as oxidative stress inflammation and endocrine activity, while stating that direct links to human disease require more research.
I understand. I will draft a proposal with realistic exposure levels and a mix of pristine and environmentally aged particles and send it to you.
Good. And remember, use realistic exposure levels in experiments so results can be translated to human risk assessment.
Listen again to part of the conversation. Then answer the question.
Use realistic exposure levels in experiments so results can be translated to human risk assessment.
This concludes the conversation portion. For more TOEFL practice tests and study materials, visit Spinta.org.